So I will be talking about the first two chapters of my PhD thesis. And you'll notice I've changed the title a little bit there. So the first chapter is dealing with, as the title would suggest, vigor in fertilized lodgepole pine trees. And we're looking at carbohydrates as a way to assess vigor. And in the second part of this talk, I'm going to talk about some work we did that looks at what happens if the mountain pine beetle overcome fertilized trees. Are they going to do better in terms of their overwinter survival in fertilized trees relative to unfertilized trees? I should also mention that for all of this work, I am supervised by Vic Liefers and Nadir Abilgin, but we also collaborated with Simon Landhauser in the Department of Renewable Resources. So since, as far as I know, since in about the 70s and 80s, there has been some interest in managing landscapes in order to manage mountain pine beetle populations. And one of the main sort of things that I have found in the literature as a means to do this is fertilization and thinning. And there was a fairly influential paper in 1985 written by Waring and Pittman. And in it, they suggested that if you fertilize and thin, you are able to increase the vigor of trees. And they used a vigor index to measure this. And so this is basically how quickly the tree is growing radially. And so relative to its leaf area. And so if you fertilize and thin to the right amount, you can increase this vigor index, and if you increase it to a certain threshold, you can make your trees less susceptible to mountain pine beetle. And so this is a pretty appealing idea to land managers. Not only can you increase the growth and yield of your managed stands, but you can also make them less susceptible. But to be honest, when I first started out, it seemed like it was a little bit too good to be true. So not only do you have your trees that are growing at a fairly maximal rate for the environment, but they are also defending extremely well. And so we designed our first study to look at what we consider to be a very good measure of tree vigor, which is carbohydrate reserves. I'm going to get into that. And as I mentioned in our second study, we look at mountain pine beetle survival in fertilized bolts. So throughout the talk, I'm going to be talking quite a bit about carbohydrates in trees, and I've already mentioned that I think they're a very important measure of vigor. You can see from this very simple diagram that you have the photosynthesis that's happening, which is converting sun energy into sugars, which are a mobile form of carbohydrates. The tree can move them to areas where they are needed, and eventually some of those carbohydrates, some of those sugars, end up as starches, or they're immobilized in different parts of the plant. And we know that those sugars and starches are used in the defense reactions of trees, and so they are important. And they are the underlying kind of mechanistic principle for why a vigorous tree defends better. From this diagram, you can also see that we tend to think of the overall flow of these carbohydrates in trees as going down. So they are produced in the crown, and then they are moved down to different parts of the tree where they are needed. Sinks in the stem where the tree is growing radially, or maintenance sinks, as well as in the roots. And so as I mentioned before, the idea of vigor is kind of predicated on carbohydrates and this sort of economic perception of how a tree will fund its defense reactions. And so it's economic in that if a tree has more money coming into the bank than it has going out, then it can use that surplus to defend itself. And this is sort of the idea underlying the carbon allocation hierarchy hypothesis, which was put forward by Waring and Pittman to explain why trees with large radial increments tended to defend better. But there are a number of other kind of competing defense theories out there, and some of them suggest, as in this lower diagram, that you can have growth and defense competing for the same carbohydrate pool. And so if you want to know, if you want to get at which trees are going to defend well, it might be not such a great idea to look only at growth, because growth might be actually reducing the carbohydrate pool available for defense. And so to get at this idea of whether and how growth relates to the carbohydrate supply in trees, we were fortunate to work with the Foothills Growth and Yield Association, and we used some of their fertilized and thin sites. And so there were 10 sites, or 8 sites that we chose of theirs, and those were situated in the foothills of the Rocky Mountains, from about Grand Prairie down to about Sundre. And as you can see in each site, there were 4 plots that we chose. The first was a control, a fertilized plot that was fertilized with a blend fertilizer that was specifically formulated for the soil and deficiencies in that those areas. We have a thinned treatment where the stand was thinned from below to a density of 2,500 coniferous stems per hectare. And we have fertilized and thin, where both treatments were combined. And so this is a 2x2 factorial design. And so some of our results, these are some early results that I haven't bothered to reformat, but I'd like to point out some specific things here. Here we have the treatments along the x-axis. So we have control, fertilized, fertilized and thinned only. And we have growth increment area. So this is the when we went out to those sites, we sampled a number of tree variables, including diameter at breast height, height to live crown, total height of the tree, and we took a root sample and looked at carbohydrates in that root. I will get to in a second why we consider root carbohydrates to be a particularly important indicator of the carbohydrate supply in the tree. But anyway, so these are our treatments along the x-axis, growth increment area. So those trees that were in those fertilized and thin sites actually had increased growth increment area, as you would expect. But the root starches, the percent of reserves in the roots of those trees, was very low. So as I mentioned, we took a few tree measurements that we were trying to relate to or use to predict the carbohydrate status of the tree, and the most important of those was height to live crown. So I will now sort of explain why we think that root carbohydrates are a good indicator of the reserves of the tree. As I mentioned before, we think that the flow of basically the direction of transport of sugars is downward in the stem. And so the carbohydrates that actually end up down here in the roots are the ones that have survived this gauntlet of the stem. They have not been spent or used for growth, and they haven't been used for maintenance or defense. So those are the ones that actually made it. And so a few things to point out here then. We see a nice nonlinear decreasing relationship between height to live crown and root starch concentrations on the y-axis. And also, very noticeably, you see that the unfertilized plots, those open circles, are higher in root starch concentrations than the fertilized plots. So from that study, we concluded that at least in the first year after fertilization, you tend to see a decrease in the availability of carbohydrates that the tree might use to defend. We didn't, in that study, measure directly any defense-related characteristics, but we were doing that in my later work. So we know that the fertilization might potentially actually make the tree less happy in terms of how many carbohydrates it has. I'd like to now move on to the next question we had, which is, let's say that fertilization does work. It does make trees less susceptible to mortality from mountain pine beetle. Well, in the event of a very extreme outbreak, some of those trees are going to succumb. Well, how will the mountain pine beetle do in those fertilized trees? And so in this study, we had what we considered, it was an older stand. It was over 100 years old in Nojack, Alberta. And our questions were the one that I just indicated to you, but we also wanted to know if you rear beetles and fertilized bolts, maybe they will do better in cooler conditions. They will have higher fat reserves because of growing in higher nutrient flow. And so we designed the study to also address that question, whether higher nutrient levels might increase survival in cool conditions. So this a little bit about the layout of the study. We selected 30 trees in this site in Nojak that you can see right here. And in the spring of 2009, in May, we fertilized those. We just fertilized around the radius around the bowl of each tree. We cut those trees down in late August or mid-August, and we cut 4 bolts from each tree and trucked those out. This was a whole lot of work. Not only we had quads here, but when we moved them into the sites that they were eventually going to go into, we did it by hand. But the reason we sectioned 4 bolts is because 2 of those bolts were going to be infested with mountain pine beetle, and 2 of them were going to be infested with the fungal symbionts of mountain pine beetle. And one of each of those pairs would go up to Grand Prairie, where we knew that the winter temperatures were about 5 degrees cooler on average, and one of those pairs would go down to the Crow's Nest Pass. So, as I mentioned, we sectioned them into 4 bolts. We brought those out of the field and into the laboratory where we inoculated each beetle-infested bolt with 4 pairs of mountain pine beetle, 4 mating pairs. We also inoculated the fungal-infected bolts once with one of the major fungal symbionts of mountain pine beetle. So we used Grossmania clavigera, Leptographium longiclavatum, and Ophiostoma montium. So once we had the bolts inoculated and all infested up, we brought them out to the field. So this is out in the site north of Grand Prairie. And we placed them in mountain pine beetle infested stands. We didn't want to risk releasing mountain pine beetle into areas where they were not already, but we also screened them in a metal screen. So the ones that you see here hanging from metal screen are mountain pine beetle infested. This is a fungal infected bowl. And so the stands looked quite surreal. We hung them this way actually in order to have the bolts above the snow line. So they were hung about 1.5 to 2 meters from the ground in order to have them above the snow line. This here is a temperature logger, so at each of our sites we could monitor the winter temperatures as well. And so those bolts were put out there in late September. They overwintered in the winter of 2010. And in the spring of 2011, in May, we went out and grabbed those bolts, brought them back to the laboratory, and we peeled back the bark to see how the beetles were doing. And one of the measurements we took was the galleries, larval galleries and pupal chambers. So from this we could get an idea of how many larvae were produced and how many made it to the pupal stage. And so you can see here, this is a typical gallery picture, and you can count out the number of larval galleries coming off of the maternal gallery as well as the pupal chambers. And so that's what this data is here. So we have larval galleries on the left here. This is at the northern site, pupal chambers here, larval galleries at the Crow's Nest Pass site, and pupal chambers. And so what you'll notice right away is that in general in our fertilized bolts, the numbers of galleries and pupal chambers were consistently higher. These are 95% confidence intervals, these are means. And you can see that for the most part they do overlap. And so those wouldn't be considered significant differences, except for here. So in at the northern site, in our fertilized bolts, we had many more pupal chambers than in unfertilized bolts. And so what we think is happening there is that the fertilization may have sped up the development or permitted more of those larvae to reach that pupal stage. This is another set of data that we collected, so this is slightly different variation. We were counting basically the number of living individuals total number of living individuals extracted from those different bolts. And so draw your attention first to this on the left. We have control and fertilized in the south, and average number of individuals per bolt. And so we collected a mean of 10 in fertilized bolts in the south, whereas we collected less than 4 in fertilized bolts, or control bolts in the south. And so that's more than double. You can also see that most of those individuals, those living individuals that we did collect, this was in June, around early June, were larvae, with a few pupae here and very few tenoral adults. Now, it's been a little bit, the part of the graph that I've kind of skipped is the problematic part, which is this here. So based on this, we have more pupal chambers in fertilized bolts, around 20. We would expect to have more individuals in the north as well surviving to be collected by us. And this isn't what we saw. We collected very few and often none, no living individuals from our bolts in the north. From our temperature loggers, we know that there was a spring drop down to about minus 20 degrees, but we're really not sure what this cause of mortality is. We don't know if that was it, but there was something that happened between the time that they built pupal chambers and the time that we sampled, where we had really high mortality, and we'd really like to know what that is, but we haven't figured it out. So it looks like that fertilization may actually increase the ability of the beetles to survive over that winter period, at least in the southern location. So our next question is, can this be linked to the fungal symbionts? So does fertilization increase the growth rate of the fungal symbionts of mountain pine beetle? And the answer is an unequivocal no. So this is Grossmania clavigera, Leptographium longiclavatum, and Augustova montium. This is the natural logarithm of the area colonized, which we measured on the basically the surface of the vascular camium. And you can see that in those fertilized bolts, it wasn't consistently higher in the south or in the north. So the area colonized was not higher. But what we did find, we took a sample of phloem from those areas that were colonized by the fungal associates, and we compared it to basically phloem from the same trees that was not colonized. And this is in agreement with the literature. We find that these fungi, especially Grossmania clavigera and Leptographium longiclavatum, are very good concentrators of nutrients in the phloem. So so I'll just go a little bit more into detail there. We have our control bolts and our fertilized bolts with no fungal infection. This is Grossmania clavigera, so control fungal infection. So it's quite a bit higher. And the same for Leptographium longiclavatum, and to a certain extent the same for Augustova montium. And so what we think is happening here is that the hyphae of these fungal symbionts move into areas of the tree besides the phloem, and they likely are sort of concentrating the available nutrients that were in the soil or in the other tissues of the plant, and bringing them over into the phloem. And so this is a sort of it's already been in the literature, is already sort of a hypothesis for one of the main reasons what the fungal symbionts are symbionts. They kind of increase the nutritional quality of the phloem for the beetles. And so the notable thing here is that this is especially so in fertilized bolts. So they're very good at concentrating nutrients in fertilized bolts. And so the implications of this study, so fertilization we know increases phloem nitrogen. This is one of the things that we measured. It can be further amplified by fungal associates of the mountain pine beetle. And this may lead to, we emphasize may, lead to increased survival and development rates in our beetles, especially in beetles that are in mild conditions. And so some overall management implications, fertilization reduces carbohydrate reserves in pine trees. We're going to try to tie that more to actually defensive reactions, but the other is that it has the potential to increase brood productivity. So if we are to draw back and look at fertilization and thinning as management strategies, I know that there has been some work on thinning in BC, and it has held some promise in terms of reducing mortality due to mountain pine beetle. But from our studies here, we think that fertilization may be a little bit more of a risky strategy. So I'd like to thank a few people, Adrian Rice and Nina Hunsdorfer, Barry White, Brooks Horn, Daryl Gilday, Dave Morgan, Devin Letourneau, and Jeffrey Millett. Those people really helped us with that second study with the mountain pine beetle. We couldn't have done it without them. And then from the University of Alberta, Candace Serban, Caroline Courtois, Christine Dahl, Eckhart Marenholz, Evan Ash, Kim Stang, Lucas Valdeholen, Ryan Sherritt, and Frank Chow. Thank you.